Hello, dear student. I failed to continue my class due to network connection. From previous slide, I started again. I already discussed about the rough endoplasmic reticulum. Now, smooth endoplasmic reticulum. In lipid synthesizing cell contain lot of smooth endoplasmic reticulum, and its function is synthesis of lipid and carbohydrate metabolism. It has also another function, that is detoxification. Here rough endoplasmic reticulum and polyribosome helps in synthesis in protein. Thereafter it synthesizing materials transmitted to the Golgi complex, and from the Golgi complex you see different by passing different segment, the nutrient material are passes through outside the cell and gives nutrition to other cells. Packers and movers, Golgi complex. It is the actually act as like postman. That means it carry the different nutrient material that are secreted from smooth or rough endoplasmic reticulum. Thereafter, it stores, packages, and thereafter transport in different area. So packers and movers. Golgi apparatus or Golgi complex is a highly dynamic membrane-bound organelles that complete the post-translational modification of protein synthesizing in the rough endoplasmic reticulum, then package and address those protein to proper destination. So we can say it as postman. And histologist Camillo Golgi, who discovered it in 1898. The structure you see, it has two zones, that receptive zone and transmitting zone. Receptive zone is known as cis phase and transmitting zone is known as trans phase. Here cis phase accept the substance secreted from endoplasmic reticulum, thereafter transmit through transmitted zone by producing different vesicle. Here you see that rough endoplasmic reticulum and Golgi apparatus both are sac-like structure, different sac, and from sac, protrusion of sac, it form a large vesicle, and that vesicle contain those nutrient substance and transmitted to their destination site. Hydrolytic unit of the cell, that is lysosome. Here showing a foreign particle or bacteria engulfed by a vesicle-like substance, that is lysosome. When bacteria enter into the cell, lysosome goes to the bacterial site and engulf it. Thereafter, by help of hydrolytic enzyme, it destroy it, or it exists that bacteria or foreign substance. And the lysosome has more than 40 different hydrolytic enzymes, and that has a highly great phagocytic activity. It type is two type, that is primary lysosome and secondary lysosome. And primary is inactive lysosome, secondary lysosome is active or heterolysosome. And secretory granules actually originated from Golgi apparatus, are found in cells that store a product until released from the cells by exocytosis. Secretory granules, I already described that Golgi apparatus transzone produce a few protrusion-like sac, and that sac ultimately separated from the Golgi apparatus and form vesicles, that also known as secretory granules. Here lysosome producing function, see that different bacteria when enter into the cell, the lysosome engulf the bacteria, and it acts as a phagolysosome and it destroy it. Here lysosome has a different function, that means autophagy. Autophagy means the process by which lysosome remove excess or non-functional organelles of the cells is called autophagy. That means when cells like mitochondria or other cell of the cell cytoplasm when remains non-functioning, or old cells are need to destroy or exit, then lysosome can engulf the that organism and non-functional organism, and can be destroy or vanish these cells, or drink or eat those cells. So it is termed as autophagy. That means own cells, organisms can be destroyed or uh, eat or vanished by lysosome. It also need due to newer cell organisms are formed and perform their function, and old cells need to vanish. So autophagy performed by lysosome. It's an important question for exam. Thereafter, it is also known as scavenger of cell. Scavenger means it can remove the extra organisms or foreign materials, and it exit from those cells. So it is termed as scavenger also. And sometimes it produce few residual body within it, because of indigested material is retained within that cell and vacuolar compartment, known as residual body. So function, degradation of denatured and non-functional polypeptide, degrade protein no longer needed by the cells, restrict an activity of a specific protein for a specific period, and main function is autophagy or act as a scavenger. Those are the main function. Cartoon showing that mitochondria, power generators of the cell. Mitochondria are membrane-bound organelles which supplies energy for most cellular activities. That means we perform function by help of energy that comes from mitochondria. That energy is termed as ATP, or adenosine triphosphate, high energy phosphate. Where ATP are need actually our whole body need ATP, but main sites are, that means liver, cardiac muscles, cells of the kidney tube. Those three organs actually perform their function continuously and need lot of ATP. So mitochondria, huge amount of mitochondria present in those area or organ, and its structure is it also membranous organelles, and by layer present in the outside, that is outer layer, inner layer, and inner layer forms different infolding, that means inner fold folding present, and term as crista, and a few intermembranous space present in between two membrane, and innermost compartment is known as matrix. Actually, fire is representative of ATP. That means powerhouse of energy. Function, we know that storehouse of energy, and synthesize ATP by oxidative phosphorylation. Peroxisome. Peroxisomes are spherical membranous organelles, and because of we know that different enzymes names are like this, and peroxisome produce also few enzymes that degrade hydrogen peroxide, and present almost in all cells but abundant in hepatocytes and kidney cells. Function, by help of those enzymes they can degrade the different foreign particles or uh, inactive different toxic in materials like drug. Now centriole and centrosome. When two centriole remain in a right angle, that is termed as centrosome. But when single centriole we discussed, this is centriole. And centriole is the microtubule organizing center for the mitotic spindle and consists of a paired centriole. that is centrosome. And centriole, how it formed? It formed actually by microtubule. Different microtubules are arranged in round shape and form centriole. And during mitosis process, you see that mitotic spindle form, and this is produced by centrosome, and centrosome are divided into two centriole and separated into opposite pole, and goes to opposite pole and enter into new dividing cells, that means daughter cells. It remains near the nucleus. Function during mitosis, centrosome induce the formation of mitotic spindle by migrating to the opposite pole. Now nucleus, that is most centrally present of the cells, and it consists of outer nuclear membrane, nuclear pore, thereafter in a central nucleolus, nucleoplasm, and chromosomes or chromatin. Nuclear membrane is a selective barrier of cytoplasm, and that cytoplasm is termed there in nucleoplasm, and it composed of two layers. Different nuclear pore present because of selective passage of substance across the membrane. Here showing nuclear membrane, nuclear envelope, that is has nuclear pore and few cytoplasm, that is termed as nucleoplasm, centrally nucleolus, and chromatins or chromatid chromosomes are attached with it. Chromatin is non-dividing cells that are there is a network of thread-like structures called chromatin, consist of DNA and associated protein, and two types, heterochromatin and euchromatin. When it is looks like light fine, that is termed as euchromatin, and when it is dark and dense stain, that is termed as heterochromatin. And here DNA, it consists of DNA, and in nucleolus it is the site of synthesis of ribosomal RNA. DNA bihelix structure. Now cytoskeleton. Cytoskeleton are microtubule, microfilament, intermediate filament, different non-membranous organelles. Microtubules are located in the cytoplasm and radiating from the centrosome. Because of, we already told that the centrosome or centrioles are actually structurally formed by microtubules. Different microtubules are arranged and surround area and form centriole, and two centriole form as pair in right angle, that is termed as centrosome. And microtubule also present in the cilia and flagella, because when a cell need to move or remains adherent, that means maintain, it actually maintains cell shape, and other functional movement as like cilia and flagella, and provide track for movement of organelles and chromosomes. Here showing movement of a cell by help of microfilament. Microfilament means actin filament are located beneath the cell membrane. Function, contract and move the cell, change cell shape, cytoplasmic transport. Intermediate filament. Intermediate filament are located throughout the cytoplasm inside the nuclear envelope also, and function, strengthen the cell, maintain cell shape, and maintain the nuclear shape. Now inclusions. Inclusions are aggregation of specific types of molecule. They have little or no metabolic activity, contains accumulated metabolic substance, and are not enclosed by membrane. Examples are fat droplet present in the adipocyte, liver cell, glycogen granules present in mainly liver cell, lipofuscin like pigment, that is a brown pigment found in the neuron cardiac muscles. So important short note, inclusion body. that frequently come in exam. And another question also, difference between the organisms and inclusion. Organism means it has membranous or non-membranous organisms, but and highly metabolic substances are organisms, but inclusions are non or little metabolic activity, and those are actually contain accumulated metabolites. And examples you can mention fat droplet, glycogen granule, and lipofuscin. So thank you all. See you later.